Yeah, just as you be on Twitter. Not yet.
A number of different things. So,
we need to have that information, like the website. First of all, I mean, I need to. Yes, maybe that one would be nice.
No, I'm sorry. You have to get to some comment. And it must for this. Everything
It's more But I thought that it's like still maybe no, yeah, maybe like it's going to transition a bit. Yeah. Well, I know it's starting up something new, it's something that's we can talk more in the breaks, maybe.
To run home.
And mine still hasn't seen it. Sorry.
Only by probably stuff, yes, because it gets deleted from the created. Yeah, it's some. Like masks. Sorry, I must oh, about this.
Hello everyone, good morning. Okay. All good? You get the coffee, breakfast?
Yeah.
Cool, so we're happy to have you here. We're a bunch of enthusiasts from in genomics, coding, Node, JavaScript, trying to make genomics data and all these technologies more easier accessible. So we are from DNAdigest, Node, Repository. Bruno started from the book lab, but Queen's Mary, Campus London is hosting us today here, and we have many friends from the community. So before we start, I want to thank Campus London for hosting us here. So I guess and I guess many of you, or some of you know about Campus London. So it offers free events hosting, and it's built for the community. It's supported by Google for entrepreneurs, and it has a mission to provide space for entrepreneurs to learn, connect, to build good startups. And the membership to the campus is free, so you can just go on the website, subscribe, you can access. It's a very cool cafeteria downstairs. For the new ones. And it has places all around the world, so it has Madrid, Sao Paulo, Seoul, Madrid, Warsaw. Yeah, also many thanks to Repository for making this event possible, so for offering the lunch and for all the volunteers. So everyone wearing a blue t-shirt, so maybe blue, yeah, sorry. A yellow t-shirt, so maybe you guys can stand out. So, I mean, we're quite visible, right? Yeah, so just ask around. We'll try to help wherever possible. Health and safety, fire exit, that door there behind, going down to the cafeteria. So on the right, down the fire stairs, down to the cafeteria, and the place where the security will take care of that. There is a small kitchen behind, so you can, and the toilets are also just behind here, to the door on the left. There are some toilets. Cool. Let's see. So the network. So for the ones that are not connected, please do so. Couple of administrative things. So in order to make this work, we have a lot of information and we prepare a lot of things on GitHub. So there is a GitHub organization called Hack. So if you didn't subscribe already, please go to hack.bironode.io and follow the process there. So it's basically, if you have a GitHub account, it will add you to the organization. If you don't have a GitHub account, you just need to make a GitHub account. It takes 10-15 seconds. And then you follow like hack.bironode.io and then you're added to the organization. Most of the documentation and most of the content will be shared on Bironode Hack, so github.com Bironode Hack. And the communication between us will be on Gitter. So the same, once you have the GitHub account, just follow this link, join the Gitter chapter. Please feel free to tweet as much as you can. So Bironode Hack is the hashtag for today. Genomics, because we're working genomic data, Node.js, like that. Campus London here. So, any questions until now? No? Cool. And basically, the program for the day, you will find it on Gitter. So it's essential for you guys to follow those things because there is a lot of information that we found in there. So quickly, the program for the day will tell you a bit about DNAdigest, so Fiona, which is the founder and CEO of DNAdigest, and of course, they will tell you why are we here, why do we try to work with within the genomic space. Then Bruno will tell you about how he got to build Bironode, the open source community. Then we'll tell you a bit another 15-20 minute presentation on how do we see what's the landscape of DNA data, and we'll do a bit of tutorial on streams, pipes, all cool Node.js stuff, and then we should hopefully end by 11:45. We can start grouping in teams and start working on some projects. So that's roughly cool. Fiona.
Okay, so I'll just give you a quick intro to DNAdigest. So this is just one of the many events that we do. We do around 4 events a year. But if you never met DNAdigest before, you might wonder why is DNAdigest doing this kind of event. So DNAdigest is a UK registered charity where we support best practices in data sharing in genomics research. So we've existed for a bit over 3 years now, 3.5 years about, and since 2 years ago we spun out Repositive as a company to help build tools to facilitate data sharing in the genomics community. But the whole mission behind DNAdigest and all the events that we do, it's all about enabling data sharing for research and making data more accessible and available. So that's the kind of data that researchers need for genomics research. And I know that not everyone in the room has genomics or bioinformatics background, so just a few words to give the basic context of what we're doing. So the reason why researchers want genomics data, and when I say genomics data, it's really DNA data. It's the DNA that's in all of us, which is the blueprint for what kind of genes, what kind of proteins are expressed in your body, and I say including the basics for all the functions of the cell. The advent of DNA sequencing and these technologies that lets you look at a whole genome and see all the base pairs, the ATGCs, all these structures that within the DNA. That's what we're using in research today to find out both how the cells work, like what are the functions of the cell, what do genes do, what genes are important for what. But it also means that these are the same kind of, this kind of information is the same kind of information we use to study cancer and genetic diseases to find out when something goes wrong. So you need to look at all the sequences of DNA to find out what are the mutations that are causing the cancer, what are the mutations that are causing a protein not to work. Or if you want to find out what's the relation between different species, you do that by comparing the DNA of them. So there's lots of uses of this type of data, and there's lots of data out there in the world, but unfortunately because research community tends to be a bit behind the rest of the world, so although everybody else is internet connected and easy to share and access different types of data, in the research world, the DNA data and genomics data is really hard to get by. There's more infrastructure needed around how to make it easy to find and access, but there's also a lot of culture change needed about spending the effort to making it easy to find and access, which needs a bit of bringing the awareness to researchers about why it's important to make the data available. So what we do in DNAdigest is that we run these events and workshops to raise awareness in the community. So it's both the research community but also the public about why is DNA data important, why it's important to get access to genomics data for research, what is the impact of doing so and what's the impact of not doing so, so we know how important it is. So this will give you a bit of a taste of the last 3 events before this one. We did a symposium last year about the challenges of data sharing, where we had voices from both the research community, from publishers, from funding bodies, and from startups in genomics to talk about what they can and cannot do with regards to data sharing. We talks from the Wellcome Trust, we talks from Trust Biology, some of the voices that in this community really affect. We had a hackday back in December last year from the content mine, where we were looking at how you could use text mining tools from the content mine to extract information from papers, and we use that to find out which papers are mentioning data and which data is available, which data is not available. And a couple of months ago, we did a workshop at the University of Cambridge on what tools are available to find and access genomics data. So those were mainly biologists and clinicians that wanted to know what tools are available. So not a hackday as such, but using tools that are already existing. But what we're doing today is much more hands-on coding style. And it's BioNode hack, and there's the hashtag for you if you forgot it already, but I'll remind you a couple of times, which is all about how we can use the BioNode framework to make open source tools to make it easier to access data. And Bruno will tell you much more about that. This is Bruno, by the way. He's the one in the black t-shirt, just to be different. So the aim of today is to have a community learning experience, to share what are the tools and BioNodes that can be used for this purpose, and share the best practices and share a lot of information between you guys, because a lot of you have a programming experience, a lot of you have some bioinformatics or genomics experience, so the idea is to shuffle a bit around in groups so there can be some knowledge exchange going on, you can all learn from each other and make the most out of it. And also, part of this day is also to have fun. So please enjoy and make sure you have a tea and coffee, and you make sure there's a nice lunch for you as well. And if there's any problems, of course, do come and ask. And if you, after this event, you're interested in more events from DNAdigest, you should check out our website at DNAdigest.org. You can sign up for a newsletter or follow us on Twitter to see what's happening. And then we're very happy to welcome you to future events. So I think I'll pass from here on to.
No, we need to change that.
Oh, then we change to Bruno?
We
Go. Bruno.
just want to do I'm going to put the slides on the GitHub channel. Is everyone on the managed to register to the organization is on GitHub channel? You know what to do.
Yes, that's your okay.
Slow. Sorry about that. So I'm just going to give a small introduction about the final project. So the overall aim of this project is to try to make modular and universal tools for bioinformatics. So what we mean by this is to try to make tools that do one thing well, try to provide highly reusable tools and code through JavaScript libraries or command line tools. Scale by using streams, because one of the problems in bioinformatics is the huge amount of data, so we want to try to take advantage of streams to process that. And finally, run everywhere, which is a great advantage of JavaScript. It would be surprised how much time scientists waste trying to get their software to work on different machines. So to do this, you're using Node.js, because it maps to what we're trying to achieve. So Node.js is highly modular and has a very open community on GitHub. And you can see that by just looking at the number of Node.js modules that are published, which is growing exponentially. Which is interesting when compared, for example, with languages that are more popular in biology, like R or Python. So even though Node.js is growing a lot in everywhere, it still has to catch up in biology. And the other really cool thing about Node.js is that it provides a native implementation of streams. So after this talk, I'll give a brief introduction about Node.js streams, so I'll explain that in more detail there. And finally, you can run JavaScript on the command line and on the browser. So for example, the letters are probably too small, but here I have a terminal that has just it's parsing a file format from bioinformatics, a format called FASTA. So here I'm just running the test to make sure that my browser is working. So we use read file by passing file. So a FASTA file format is something that looks like this. You have an identifier in a genomic sequence. And what I'm doing is just converting it to a JSON object. And then I'm doing a bunch of other fancy stuff. The cool thing is that then I can run exactly the same code without modifying it
on the browser. So it's the exact same test running here on the browser console. And the only thing I had to do is, because I'm using the Node.js API, I had to use this tool called Browserify, which will allow me to use the same API on the browser. But I think this is very powerful to be able to write code once and then run it everywhere. But people might say, well, but maybe JavaScript is not the best language for highly intensive scientific computation, for example. What about C++? Something like that. The cool thing is you can also combine C++ very nicely with JavaScript. So you can run C code in the Google V8 engine, so inside Node, or you can transcribe it to JavaScript and run it on the browser. So there are a bunch of projects like this out there, which allow you to combine a language like C and JavaScript. So with this, you can really build software that is cross-platform. So I think it's a very nice step to be working on. So by what happened with my PhD, so I'm in my final year of my PhD, and when I started, I got involved in some biological web projects. And so one of them was this one. So this is like a genome browser. So what you have here are the genes, and the user is editing the annotation of those genes. So this happens in the browser. So it's this part is all JavaScript. But this software that I got involved has like the front end was JavaScript, but the back end was Java. And because of this, it didn't scale, because a lot of things that could have been done on the client side or things on a server without any good reason for it. So what I started doing was to try convert some of the functions to JavaScript to be able to do some things on the browser. That's how the first binary model happened, which is a very basic model. It's binary sec, so it does things like just checking if sequence is DNA or if it's RNA. So basically just pattern matching, just regular expression. But the thing is, there was no library doing this. So if you go on GitHub and try to put, for example, this one just do a reverse of the sequence, so this one does a reverse and a complement, so like basic stuff. But everyone just keeps re-implementing these things, so no one is using a library, which means it's very easy to make small mistakes because you're not looking at the full scope of the problem. So it's actually better if you just combine all knowledge on a few tools. So that was how I started coding JavaScript for bioinformatics. But then the other problem I had is, for my PhD, I had to fetch terabytes of data from online resources. So JavaScript is also very nice for that, to fetch data online. So for my PhD thesis, I wanted to compare social species like ants and bees and termites, compare their genetic diversity with solitary species, like, for example, cockroach. So you would expect the hypothesis that the social species have a lower diversity than the solitary species. So, but the problem is that there's a maze of data sources out there. And after looking into all this for a while, I ended up just using one database called NCBI, which is where most of the genomes are stored. And so from a quick query on that database, I found that I could potentially use 167 species. And so I wanted to compare the blue ones with the red ones. But the problem is, from all these species, I didn't know which one I could actually use. Because the method I was using has some restrictions according to the sample on which the DNA came from. So I need to get all the information about all these genomes before I can make a decision. And so that's why I created the model bioinformatics guide to query this online resource. So I'm just going to give an example of how this model can work. So for example, if I wanted to figure out where the entity server, where the genome is stored, this is genome for an answer, this is raw data, but I don't know where this information is, so I need to very difficult to figure it out. So when I query the database, I get the JSON object like this from Bio.NCBI. So this is what Bio.NCBI provides me when you do this specific query. So I'm going to show you how this works. So the one I was interested in just the genomic data, just the DNA. So in JavaScript, you require the binode module. And this module is actually a meta module because it will require a bunch of other binode modules. So you can use them as separate modules if you just require one library, or you can use this one and fetch it everything. Once I have that one, I can fetch the data using what's common in JavaScript called a callback pattern. So I do something like this: bio.ncbi URLs. I'm looking for the genome, so the genome assembly. So I look into the assembly database for all the ants, for example. And once I get there, it calls a callback that will give me an object with all the URLs. Then I can console log, for example, the first one and get just the genome because I don't care about the other URLs. But the thing with this is that I'll have to wait for all the data to get fetched. Before that, I won't get any result. And actually, I could be doing something useful while I'm fetching all these species. So one alternative way to do it is to use the event pattern. So because Bio.NCBI uses streams, what I can do is I can do the same query, but now I'll attach an event. So on data, when I receive a data event, I run this function to print the genome URL. So now I'll be printing URLs as I get for each species from Bio.NCBI. And the cool thing is that there's still a lot of boilerplate code here. So I can actually do this using the pipe pattern. So here I'm requiring only the Bio.NCBI module. I'm requiring a JSON parser. And what I'm doing is I do the same query, but now I pipe to the JSON parser to convert the JavaScript object into a string. And then I pipe directly to the terminal. So this is very, very powerful because this starts looking like what we use a lot in bioinformatics, Unix pipes. So it's very easy from this to go to this. So I can also use the binode library as a command line tool. I can do exactly the same thing that I showed you on the command line, and I can pipe to a JSON parser, and then I can get, for example, the first result. This allows me to use it without using JavaScript, if I want to, and to combine it with other existing tools. So after that, I used binode to fetch all the metadata I could, and actually figure out, because of several reasons, that I could only use like a very small subset of this genome. So this saved me a lot of time. Otherwise, I would have probably to look at all the papers and all the links and all the database, figure out if I could use that. So actually, the raw data from my PhD compressed now that I can use is 7 terabytes. So the other reason why I started doing all this is that I had to also build a very complicated bioinformatics pipeline. So as I showed you, just to fetch the data, I had to do a lot of stuff. And so with streams, it's very nice to create pipelines. So a pipeline is basically you have a visible stream, like you fetch something from the web, you can pipe to a transform stream, do something with it, and then you can write to the terminal or to a file. So this is an example of the pipeline I used to filter to get all the metadata for the genomes I needed. So what's happening here at the beginning, I'm querying for the raw data set. But then once I get that metadata, each branch is querying the sample metadata, the paper metadata, the project metadata, the reference genome metadata. So I'm querying all these things at the same time while I'm fetching one object from here, like each species. While that's happening, I'm doing all this and I'm storing everything into the database so I can then filter it all. But you can see it's starting to look very complicated. And the other thing is also, this work is for metadata processing, but actually to do the analysis, I also have to come up with some crazy pipelines. So on this one here, I'm just processing the reference genome and the raw data through a series of steps, so I can then compare the raw data with the reference and look for variance, basically look for difference. So I have to use all this software and combine all them together and process the output of one into the input of the other. So I think Node.js is very nice for this kind of pipeline handling. So I'm going to give you an example of what the pipeline would look like for the metadata. So here, I'm doing a search for the SRA. So SRA is a small reads archive. So it's an archive of raw data, and I'm looking for this species. Now, the cool thing I can do with Node.js Streams is I can have a pass-through stream, which I'll explain in the second part of the talk. But from this stream, I'm fetching data here and I'm storing into a database. But the cool thing now is that the data that came through here, I can invite it to something else. So I can extract something from that object, like a sample ID, and I can do another query for the sample. And then I can store that into a database. Then I can have other stores that use the same data that came here to do other stuff. So that's how I did that crazy workflow that you saw before. And the cool thing is, if I attach to each of these steps a counter, just to see data passing through, I can see another nice property of streams, which is backpressure. So Node.js streams handle themselves, the flow of data. So here, as I'm fetching the reads, around like 50 data sets per second, so this is querying the database online. I'm fetching all the other metadata on other databases related to these reads. So what you could see was that the numbers were incrementing at different speeds, and everything was regulating itself without anything crashing or losing data. So I don't have to worry if a request is taking too long and might drop because the stream will just wait for it. And so, because of all these crazy workflows and pipelines, things we were doing, we actually got to Google some notebook student now that is trying to implement a proper bi-node workflow engine so that you can combine Node.js modules and command line tools and do this kind of crazy pipeline processing. And although the aim is for bioinformatics, once it's done, it should probably work with any use case, like physics or data analytics. So finally, when I started all this, I just did it because I thought it was a good idea to put my code online, to open source it. I didn't see any disadvantage of it. And now it has taken off, and there's already a lot of people involved. We're doing this hackathon, and we have people from other projects contributing to this project, and we exchange ideas, which I think is very useful. And that's it. Thank you.
Any questions? Feel free to interrupt us, ask us. Yeah? Don't be shy.
Hi everyone, I'm Charlotte. So I work for Repositive, but I also volunteer with DNAdigest. And I'm here to kind of talk to you more about genomic data and genomic data landscapes out there and kind of introduce what's out there and what we're doing. So during the day, if you have questions about genomics data, come to me or Richard or Fiona. We say on our labels what we're good at. If you're interested more in like no questions, there are other people in yellow that say that you can ask them. So different expertises. Okay, so you may or may not have heard of Repositive. I know you must have done, but if you want to know what it stands for, it's a positive experience of accessing human genomic data. So briefly, even though Fiona kind of covered this, but kind of what is genomic data? Kind of very overview concept. Basically, genomics is a discipline as part of genetics, and the genome is like the complete set of DNA within an individual organism. And so genomics is basically the study of the genome, which can involve multiple steps. And here I've mentioned just a few, but basically array, NGS, and bioinformatics, basically because you need to sequence, assemble, and analyze that genome to understand it. So genomic data is basically the data that comes from the genome. Genomics is the study of that data. So there's a lot of data out there. We actually think we need to update this now. So mid-2015, there's at least 80 petabytes of sequence data being made, and that's increasing exponentially. So there's a huge amount of data out there. However, only about half a petabyte of that data is actually available in the public forum. So there's a huge amount of underutilized data that could be being used to help medical research, further understanding of the human genome. This is human. Okay, so what's the problem? Firstly, the data is very fragmented. It's being made all over the world by different people and different institutions, different companies. So it's in many countries and it's siloed. So people have it on their servers in their company or on their laptop in their office or in a filing cabinet kind of thing. Like I can say for my PhD, I have a whole load of data that's sitting on a computer that's not on anywhere yet. So it's fragmented and siloed. And even if it has been put out into the world, into the public forum, it's pretty confusing. This is a very small number of places where you could look for genomics data online, should you wish to. They specialize in different types of genomic data or different organisms, and most people really only know about 5. When you start, maybe as a PhD, you will have never heard of it, and you can ask people. So you only know the ones that your colleagues haven't known about, or you Google it and come up with a few. So you can spend a lot of time searching and not finding what you're looking for. And actually, like I said, this is a very small portion. So when I started Repositive in September last year, my first 3 months, I basically focused quite a lot of my time on just trying to find the sources of genomic data out there on the internet or institutions that might have genomic data. And so that was about 6 months of work, and I managed to find 163. We're now up at like 250. So there's a huge number of places where you could find genomic data, but they're hard to find, and it took a long time, and that was basically my job. Most of you guys, that is not your job. Your job is to do other stuff, to analyze it, to understand it, not to just go hunting on the internet to know where the hell it even is. So that's like hurdle number 1, right? And so kind of what I kind of wanted to introduce you to kind of what's going on globally with this data. So this here is, and you can find this on this type of website that I've got in the corner. It's quite fun to play around on if you're interested in knowing where sequencing machines are in the world, who has which sequencing machine, or how much data they're plugging out. And so this is just where the sequencing machines are globally. So Europe and the US are big hitters, but actually every continent is now making sequence data. And they have a reasonably large number of machines, and these machines are high throughput, so they're plugging out a lot of data. And so this data is being put on repositories online, so people should be able to find it. So the yellow circles are the, so far that I know of, this is not comprehensive by any means, the data sources out there. So it's kind of mirroring. We're seeing most in Europe and US again. And these basically the data sources we know of, and they're taking the data from these machines. I'm happy to have a discussion about why or why not we have not found any in the Latin America and Africa. I have various reasons why I think that might be. If you know of any, please tell me. But there are a lot of data sources out there, and they're distributed around the world, and they're taking data from their countries or even other countries. So probably a lot of the African data I know is being plugged up to European data sources. So a lot of data is being made, and there are lots of data sources. And in case you're interested in who's paying to host this data, this is the funders that fund some of these repositories. And these are just the big hitters. I mean, after this point, there's loads of like individual ones, individual institutions, individual labs, but these are the big guys. So the NIH hosts, and this is genomics data. The NIH hosts at least 10. Broad has 8, NCBI has 9, well, EBI has 9, Wellcome has 4, and BGI has 4. So these are the big guys in kind of the genomics field. These are the guys who are funding a lot of the data being made, and they're funding a lot of places where you can store that data. And at the bottom there, EBI says it has 104 services, of which 19 are repositories. So there's a lot of places you can find genomics data, which is great, but it also, again, comes back to being fragmented and confusing. Like, you can spend a lot of time searching for that data. And then there's another layer of difficulty, and that's with data accessibility. So some of you may or may not be aware, but genomic data can be in one of two conditions, really: publicly available or under controlled access. So if it's publicly available, you can go online now, you can find it. You might have to log into a website, but you can get it today, you can download it. Then there's controlled access, and that's mostly human identifiable, possibly, data. So that'll be like clinical trial studies, and you will have to apply for access, which is a hugely arduous process and can take months, where you will have to sign up for logins, and then you have to do forms to say what you want to do the data with. That gets sent off to a committee who assess whether you fulfill the legal requirements, whether they agree with your project, whether you have the IT support to host that data. And then as long as it all works out, you get given some passwords, you get encrypted data, you unencrypt it, finally you have data on your computer, analyze it. So the restricted data is quite hard to get to, but there is quite a lot of it, and it is a very valuable resource. And so another portion of what we're trying to do is help people get access to that restricted data. And again, if you want to talk about that, feel free to talk to me afterwards. But so of the repositories that I was just talking about, actually, encouragingly, about half of them are open, half of them have restricted data, and some of them have a bit of both. Some of them have one layer that's open and then another layer which is controlled. And then a small portion of them I couldn't figure out whether or not I can get to it at all, mostly because they say they have data or they're making data, but they haven't declared what they're going to do with it once they've made it, whether they're going to restrict it or control it. So if it's hard to get to, it's probably going to be restricted, but not intentionally restricted. So that's another layer of difficulty, especially if you want to go after restricted data. But as I said, there is a huge value of data out there, and a lot of people say, "Oh, well, I'm working on this specific cohort of people or this specific disease, and there's no data relevant to me." So a lot of people kind of complain online that most genomic data is done on European ancestry. It's done in Europe or North America, and a lot of that is European ancestry. But I would beg to differ that actually, when looking out there quite hard, you do find genomic data on a huge spread of ethnicities. So this is just, I didn't dig particularly deep, but when they declare what ethnicities they're covering, and you can find most of them. So a lot of people, perhaps even are looking for data from India. Like, do you know any? Actually, I know at least 5 countries that will cover Indian data. So there's actually a lot of data out there, but again, it's like, how do you find it? I'm struggling to just find the ethnicity to match the stuff you're interested in. That's not ideal. And they also cover a lot of diseases and a lot of types of data. So again, not a fully comprehensive list. What disease you're interested in, someone will be doing sequencing data on it. Most of the repositories have a mix of everything, and then you've got cancer data being the next biggest one. Not surprisingly, a lot of people are doing sequencing in cancer, and it's already been shown to be a very good way of analysing cancer data. Everyone's doing this. Also, population. basically almost every country now has decided they want to sequence their population. So there's a lot of population studies going on out there. We're going to sequence our population. So as I said, ethnicities are now being covered. And then it kind of goes down the list. But if you have a disease of interest, almost certainly someone's doing it, or any other question. And then for the assay types, again, most of it's mixed. And then you kind of have variant data and SNP data, mostly because it's smaller, probably easier to put on, and also for studies where it's identifiable, the individual identifiable, if you're using SNPs or variant data, you can remove the capacity to identify that individual, and you get a lot of power by pooling all that information. But whole genome sequencing is not far down that list. So there's a lot of different types of data out there that you could try and analyze and try and get a bit of power with. And as I said, there's actually quite a lot of data out there. This is a long scale, otherwise it's not you see most of these. So most of the repositories are cruising between 1,000 and 10,000 samples within this. This is samples, not necessarily studies, but then some of them have a lot. And actually, interestingly, the second two, the co-op program and the biobank, are basically, they've taken a massive load of individuals and they're sequencing them. You probably haven't even heard of them. They're taking up millions of people and they want to look at them, whether they're healthy or with diseases, and they're bringing it all together. So there's a lot of samples out there, and again, you may or may not even know that it exists. But again, this problem of how do you actually deal with this information, how do you analyze this stuff? So that's your job, guys. And so the kind of places where you find data is also quite diverse. Most people are only looking at like repositories, but actually, this is of the 163, so not of our now 200 odd samples, but you've got quite a few biobanks in there. They have sequence data. They don't necessarily advertise it very well because they just don't have the capacity. It's not really within their remit. You've got a lot of companies, and there are almost certainly many more, that have data that a lot of them are quite interested in sharing with you. They might not charge you, but they are interested and have quite good data. Then there's a lot of consortiums, people coming together to try and do something powerful. A lot of repositories, basically defining repositories somewhere where you can also upload data as well as download data. This is WikiLeaks, of course, and then most universities, and this is just the ones that are like declaring. I mean, sure, every lab has got their sequence data as well, just on a server that you don't even know about. So you kind of get an idea there's a lot of data out there, and it's a bit messy. So I'm going to quickly take you through like a case study. If you were looking for some data, how you might go about it just sitting in your lab. So this is actually Bruno's case study. So this is actually what he did. And so you kind of get an idea of the struggles and why we want to do this today, why we want to find a way of getting data out of repositories in a systematic way, in a more comprehensive way than just clicking through and Googling, trying to find what you're looking for. So this is going to NCBI. So NCBI is the big NIH US repository, and it has a whole load of stuff. If you've ever heard of PubMed, that's also under NCBI. And so Bruno started by going to the genome site NCBI and search for, basically looking for anthracod or rodent sequence data. And so he searches for this, he gets 167 results, but these are actually the reference genomes for anthracod or rodents. So this is actually not what he wanted. What he wanted was sequence data. Okay, this is a lot of them, it's not actually what I'm looking for. And when you look at it, okay, so this is just looking at one of them, and SRA has 43 sequences in there. Okay, well, let's go to SRA, see what we can find there, and maybe we'll get sequence data rather than reference data, and maybe we'll find something a bit more useful. So go to SRA, searching again. Now you're getting 24,000 results for anthropods or rodents. So that's quite difficult to filter through when you're seeing that basically it's a nondescript title for each item. So that's interesting, but maybe if you click on them, you're going to get something quite useful. So clicking on one, limited amount of information, 1.1 gigabyte of sample there, but not a lot of information about that sample. Like, what have we actually got here? What are we going to get if we download this? But over here, there's some other potential places you can click. And biosample sounds quite good. That maybe that'll give you information about sample. So click on biosample information for this sample, and you get a title that tells you the species and C129, or is you want to know? What is this sample? What are you going to get from this? And no attributes. So there is basically no information here. So there's, you're constantly fighting. That was like how long process? Basically at the end would be like, good, probably haven't got any further, just very frustrated. So it's a long process and hugely frustrating, and most of the time you don't find what you're looking for. It must be out there. So basically at Repositive, our vision is one-click to human genomic data access. So that whole process that Bruno went through, typing, clicking around, storing, cut that out. Like, if you know what you want, one click, find it. I mean, that's what you can do on Amazon if you're looking for a book. That's what you can do on Expedia if you're looking for a hotel. Why can't we do that for human genomic data? There's a huge amount out there. There's information about it. Why does it have to be so confusing and fragmented? So we're building a platform. So we want to make data discoverable, so you can search, see what's out there in the world in one easy-to-access portal. We want to make the access fast. So, as I said, if it's public, leading you to the right place fast. I click it, you get it. If it's controlled access, helping you with that process, making it as quick as possible, so that you can get the data, so you can analyze it. And in the end, so you can maximize community, working together, helping each other, kind of Giraffe knows something about the data, and you can put it on a repository and help Dog know, oh, this is the data I'm looking for. So feeding back to make data discoverable. So building a community around data and helping people get it. So basically, we want to make the workflow more simple, get data access faster, so then you can go ahead and actually analyze it and make discoveries that will help the world, help medical research, not waste your time on this workflow. So you can search for data, see related results. You can discover data and then you can access it. Find colleagues, collaborators, people with similar data interests over the world. And also give feedback on that data. If you know that you've downloaded and actually the description isn't quite right and there's less samples or more samples, it's not quite right. How do you tell what repositories is? How do you change that? With this, you can build that community and feed that information back, like, actually, this is a really, really useful data set. The description is poor, but actually has this, that, and the other, or actually, this has this many samples. And so, in future, the reuse will be easier and people understand what they can get from this data. So that's what we're trying to build. And today, we want you to basically, the point of this was to understand the problem and to understand why it's important that we're trying to do what Bruno's doing and actually get past this process and get to the data that's needed. And I'm really excited to see what you guys come up with. It'll be really cool. And again, feel free to ask any of us any questions at any point. And that's it. That's me.
Okay, so next, because now I guess you start thinking about what do you mean by genomics data, what do you mean by accessing those repositories, how do we want to use Node and new data technologies. We'll go another half an hour for two tutorials. So for now we'll start showing you a bit of how you can get some streams, how you can combine these datasets and so on and so forth. So this will be kind of interactive thing. So in order for this to work, you will need to have the environment ready eventually to follow up. And Ruben will tell you about what you prepare for you. So, I mean, we do have some virtual machines. We can help you set up the environment. Hopefully, we can set it up. So, until Bruno set his computer up.
So I don't know, all of you are in the GitHub organization at the moment. Is anyone that is not there? Because if you are not there, you should have Node installed in your computer, and the environment libraries in it as well. So is there anyone that is not there yet? Please, I'd be happy to let me know. Okay, so if you are there, I'm going to share now with you in Gitter link. This link I just shared in Gitter will give you access to a terminal console. And with this one, you can, with your your GitHub username and biohack as biohack as password, you should be able to log in and have the environment ready for you to play around with this in the browser. If someone is not able to access it, let me know now. Okay. If you join the GitHub organization now, in a couple of minutes you should have also access to it. So let me know if someone is having some issue with that. Okay.
What is the login again?
Login is your GitHub username. If you have in the GitHub username uppercases, use the uppercases because it's not GitHub. And the password is biohack. So I'm going to put the password from GitHub. It will prompt you to change the password.
So it's authentication token, authorization.
You have to probably, the first, when you change the password, it asks you again for the same password. You have to, I think it's that, right? It will ask you for the current password and then you change it.
So feel free during the demo if you get stuck, go in GitHub and just ask the question and try to take it from there. So just whatever question, just fire it up. Yeah.
In that same environment, I don't know if someone is using Emacs or Beam or something like that. You have Emacs, don't install now Emacs, and you have NeoBeam, so you can play with that. And Git.
Does it seem to be charging or not?
No, it's not.
I know it's not.
No, it's not.
So, what, just off and on?
Yeah.
It needs to be green.
But if you want, I can try to go and bring the power supply. Power data.
Yeah.
Stand up.
If anyone stuck at any time, just put up your hand and then we make sure someone runs to it. Okay?
Bruno, the login is biohack, right? What's the The dog, yeah. Right, and then it's, I just wanted to do
It's just that they don't notice that I was absolutely so impressed.
If anyone is still stuck, just put up your hand and we'll come to
So,
there are two pieces of shit on every table, right? So the first link allows you to register to the organization, right? So go to hack.bionode.io, right? So go there. If you have a GitHub account, things will be like two things. If you don't have a GitHub account, you just create a GitHub account, and that's like very easy. Another whatever people say. If you are creating, so if you're new to GitHub and you create a GitHub account, then you need to go back to hack.bionode.io to register to the organization, right? Once you are part of the organization, you can see all the repositories there by going to github.com/bionode-hack. The GitHub channel is listed on the third paragraph. Yeah, and to get to the virtual machine is basically hack.bionode.io/weti. Okay, so please go.
Slash what?
weti. Yeah.
W-E-T-I.
So web TTI.
Web TTI. Thank you.
Just go on Ethernet. Yeah.
FTY.
You guys.
Sorry, that's TY. Not TI.
Why? TTY.
That's TY.
Genomics,
So
I think I'm going to start. So, what I'm going to do to start is live coding. So, I'm going to try to show kind of the process you're going to have in the afternoon to build these modules to fetch data with the streams. And so the result will be something like this. So a tool that gets data from the resource. So I'm going to start coding, and you can follow on your terminal as I'm doing it. I'm going to try to keep it very simple first. So what I'm going to do, I'm going to create a readable stream that's going to fetch data from somewhere. I'm going to use the module request to do that. So this module, I'll provide the URL and I'll fetch something. So the output from this module is going to be binary data. So in Node, it's the buffer class. It's coming in chunks of 16 KB, which is the default buffer. And then I'm going to pipe that to a writable string, which is going to be just the standard out. So I'm going to try to do just that. Let's see how it works. So first I created a folder. I created an empty file, .js. And okay, so I'm gonna need request. So I need to install it. It's already installed, I'll show how to do it. So inside the folder, you do npm install request. So you do npm install request in the folder you're going to work on, and npm will install that module in a folder called Node modules. So there's here some folder named Node modules. Another useful thing you can do is to avoid having to reinstall these dependencies all the time. When you start a new project, you do npm init. And you do enter. It's going to ask you a bunch of questions to create an configuration file. So I'm just saying yes to everything for now, because I don't care. It says yes. And so now I have a package JSON file. And that file has the configuration of my dependencies. So now I did npm install request. I could have done npm install request save. So that will save that dependency on package JSON. Which means next time, if I need to install this dependency, I can just do npm install. And it will install all dependencies on the package JSON. So that would be very helpful if you need to restart this or move to another machine. Another useful information is, so when you install something with npm, it installs in your folder, in that Node modules folder, the dependency. You can also install modules globally. So if you wanted to install binary.ncpi, you do npm install binary.ncpi-g for global. So that only means you'll be able to use binary.ncpi as a command line tool. You won't be able to use it as a library. You still need to install it locally in your folder for your project. So you only do this when you want to install something for the command line. So one thing I know is going to be useful, so I should probably install already, is JSON parser for the command line. So this one, I can do npm install JSON-g, and it will be installed globally. So it won't be a library, but it will be a command line tool that I'm going to need during this tutorial. Did everyone follow up? So I have request. So now I'm going to load that module in my code. So I do var request and equals require request. Okay, so I have request. I need to fetch data from somewhere. So if you go to the BioNode Hacks organization, you have a bunch of repositories with some information. So these might be possible modules to create in the afternoon. you can organize around multiple groups. Feel free to use another, create a new one if you want. I'm going to look into this one, the binode EVA, which is the European Variant Archive. So if I click here, there's a README, and there's already a bunch of information about it, where you can get the date, what kind of date, things like that. For now, I'm just going to take the first name, just for demonstration purposes. I'm going to copy. This one. Just going to copy this one. And now I'm going to give this to request. So usually the way you can use request is you call it, you give the URL, and then you can give a function, a callback, which will get the result, and then you can do whatever you want with the result. Actually, it's will be something like this: response, request, error. So give you an error if you have an error, and the response and request, and then you can use that. But the request module is already a string, so I'm actually going to take advantage of that and use it with the pipe method. I'm just going to do request URL, and then I'm going to pipe process just that. Hopefully that will work. So if I do node EVA, my file name, I get the JSON. So it's working. If I want to explore this JSON, figure out, because I can't really read anything here. Let's see what's going on. I can pipe the JSON tool that we just installed, and it gets nicely formatted. I can also pipe it to that. Install it. So I can see that there's a bunch of information, API version, error. The only thing I care is the response, which is an array. Inside response, there's a result, which is a bunch of studies. So that's what I actually want. So I can filter that if I want. I can do JSON, response. It's an array, and I just want the first one, and result. I get the array with just that. If I put another 0, I get the first one. So,
If you try to clone the representation first,
You guys have the display script. You go back to your yes, that's definitely better. Okay. Yeah. So guys, feel free to interrupt. So you say, can you please stop for a minute, like ask a question and so on. So I think it's good if we all try to follow this because it will help what will follow in 1 hour or so. So feel free, like if you, I mean, like whatever question you have, just ask, and we'll try to answer it, right? So try to follow to connect. Follow to be on the same page. And maybe going a bit slower.
So what we did is we installed the request module with npm install request in our folder. We created an empty file, which is this eva.js. Inside the empty file, we require the request. We're giving a URL, string, and then we're doing the Python. So we're using the request module, giving it the URL, and then using this pipe method, which I will explain better later. To pipe what I'm getting from the request directly to the standard. The URL I got was just the first one here. When you go to the binode hack, binode diva, there's a I got this one, steps to metadata. The first part. So during the afternoon, you want to make this flexible enough that you can use. So for example, here I can see this URL that I'm fetching only information for humans. So Homo sapiens and the GRCH37 is the genome reference version. So ideally, we want to be able to specify the species. But for now, I'm just hard coding everything just to keep it simple. It's the JSON parser.
It should be there.
So it's already there. You can just use that.
Sorry, but it's kind of something is not there.
Then you can do something like this: run the script and filter it. The
Web version of it. For how long have you been? Yeah. Positive, it's like years. Right. All this stuff is I think from my machine about
is everyone kind of clear? No?
JSON.
No, so if I just do JSON, I'm going to do JSON and mass.
Yeah, just
And I get this object. So it's a JSON object with several properties. And by looking at it, I see that the property response as an array. It only has one object inside. And that object has property result, which is another array. And that array has several objects with study ID and the study name. So I can use all this information, but right now, let's say I just care about the result. So that's what I'm doing when I do this. I'm filtering what I get out of the JSON. So this is nice just when you're trying to understand what these APIs give you. Try to explore the data and figure out what's relevant, what's not.
oh, it is.
So you can use modules. So here in my code, I'm not doing anything yet. I'm just piping what I get from the JSON object directly to the standard. So I'm going to try to do something fancier. So I'm going to start explaining it because you might need to install some modules. So while people are still figuring out this part, it might be helpful for others to start installing it. So this is what we did. Now, here's the solution. So you can copy paste it. If you go to the Gitter channel and you get the PDF, you have the solution here. So you can just try to catch up this way. If you have some typo in your code or something.
You just use different URL.
Well, I use the same, but what I did is I broke it into an object. So, anyway, what I did is I'm using the URL module, which comes with Node, so you don't need to install it. And then I broke, to organize my code, I broke that long URL into something more meaningful. So I have the protocol defined, I have the host, which is API, European Bioinformatics Institute. The path name, which is EVA, the database, web service, REST API, version 1, metadata, studies, list. And then I have a query, so the search, which is species equal Homo sapiens. So this way, my code is cleaner. But now I have an object, and I cannot give that to request. So what I have to do is first put it back as a URL. So I use the URL.format, and I give it an object, and I'll get back the string. And then that's the string I'll give to request. So this is just to make the code cleaner. But then I can use this object and change the query, change the path. Yes.
How do we get help for as a function? Is there a man page or something?
Yeah. Bruno, can you please submit the... Yes.
So usually to get the documentation, because all these things are on GitHub, you just go to search for this on GitHub. Or on the NPM, when you go to the NPM page, so I can show you. If I go here, NPM. Let's say I want to know how to use request. Yeah.
Help, and then maybe slide it.
I usually don't use the entrance to this. But to get if you're looking for the Node core documentation, just go to the website, to the API. So here you have everything. So if I look for URL here. URL format, I give it an object. So here's the documentation if I want to know how to, what the U object should look like. The role object has several properties. So the host name, the port, the path name, search. You can use all these with the object. So this is the core when you're using a module that comes with Node. When you're using a module from the community, you go on NPM, and what you see here is actually the GitHub window. So sometimes you can just skip and go straight to GitHub. And so here you can see how to use request. So if I wanted, for example, to write to a file, I could do something like this. Request, the URL of a picture, and then pipe to a stream, a file stream to create a file for the system. So, coming back to this, this was the solution. You can get this PDF on the GitHub channel. If you want to copy paste, I'll put also the code on the GitHub. Did everyone follow up until now? Or? Okay. So, this is not a very interesting module because it doesn't do anything that's simple download. So let's say I want to manipulate that just JSON object and do something. So I want to use a transform stream to do that. But I'm going to have a problem because I want to manipulate the JSON object, and do things like filtering or look at properties. But what I'm getting from requests is actually a buffer. So I'm getting chunks of data, not a JSON object. So I need to fix that. So what I'm going to do is use a JSON parser. So I'm going to use JSON string to parse the string that comes from request, the buffer into an object. But that won't work too, because I'm getting chunks of 16 KB. So I might get an object cut in half. So the solution to this is to use split. So split can split the buffer according to something, a character. If you just use the by default, just split, it will split by new lines. So I know the API is giving me one object per line. So I can use split to concatenate the buffer until I have one line, and then split will push downstream to the parser, which will turn that into an object. Then finally, I can use it in my transforms stream to play with it. So this is what I'm going to try to do now. The solution is on the yeah. Let's see. Okay, so I'm going to need JSON stream. So var JSON stream equals var JSON stream. Now that means I need to install it because it doesn't come with Node. So again, in my project, I do npm install, or if you want to say keyboard, you just do type. Say JSON string. Now I have JSON string installed. That's good. But I need split too. I need var split equals require split. So let's do that too. npm is clear. npm install save split. So for every dependency, you have to do this. If you do the save, then next time you just need to do npm install.
In the slides, you had split 2.
Yeah, so split 1 should work, but I think actually I should be using split 2 because the difference split 2 is using version 2 of streams. So I'll explain what is the difference between version 1, 2, and 3 later. For now, don't worry too much about it. But I really want to use version 2 or version 3.
What version of Node are you using?
6 something like that. So if you have to manage Node versions, I don't know how it's set up on the Google Node, how it's set up.
6.3 as well.
6.3, okay. But if you wanted to do on your computer, one thing you can use is the N module. So you do npm install globally N. So just N. If you're using a Mac, you can just also do brew install N. That will also work. Once you have the N command, you can do NLS. You get all the versions of Node that are available. So then you can specify by number which version you want. And you can just do N stable to get the latest stable. If you want the latest, you do N latest. So just doing that, you get stable Node.js. I find it's the easiest way to manage Node version, but there's a bunch of other tools to do the same thing. Sometimes you might require on Linux or other systems to do sudo instead of permissions. So, okay. I'm also going to need a transform stream. So I'll explain how you create So here I'm just using streams that are already made. I'm not creating anything new. I want to create a new one, do something. And I'll explain how you create streams. But for now, the easiest way is to use an abstraction model called true. So I'm going to do var true equals require true to, because it's using streams version. So you need to install this model also. It's crashing the page. If you so also, well, if you have a Mac, you can just do install and if you have Homebrew. If you don't have Homebrew, it's also very easy to install. So Homebrew is a package manager for Mac. So you just Google Homebrew. It's a one-line install. You might need to have some development tools on your Mac. So when you run this, it might ask you to install more tools, like command line tools and things like that. But if you're doing already development, you should have Xcode and all those things installed. So to install Homebrew, just copy paste this into the terminal, and you'll get Homebrew. Once you have it, you can do brew install and then you can do and stable, and you get Node on your local machine. And if you go to the discussion repository, I think there are also some instructions. Yeah, so we have here the instructions for mask, for Ubuntu. And if you know how to use Docker, you can also try to use a Docker container. But this is just a final hack discussion.
Operating jump assuming has all the
Yeah,
required modules in it. Yeah. Right. Really cheating sales is cool. We're mentioning your project, just buy
I put everything on the table, and I have a bunch of others on.
Yeah.
In the breakout, we can everything. Should I continue? And I think this
there.
I'm gonna start also. Okay, so I'm gonna try to continue. Otherwise, give you a schedule. So now that I have all these modules, I can use them. So first, the problem was I needed to get the buffer that comes from request into buffers of lines. So I'm just going to do pipe. So what comes out of request, I'm going to pipe or split, just like that. So now I have a buffer made of one line. So I can now pipe that into Johnson string. Parse method. Now I can pipe this. So I'm just going to try to see if it's working. No, it won't work because now I have an object and I cannot pipe an object to the terminal. So I would have to do Johnson string stringify to put it back as a string to pipe to the terminal. So I want to do something with this. So I'm gonna create my own string. So the way I do this, I do var my filter. It's gonna be I'm gonna use true. And in this case, I'm not going to use buffers. I'm going to use objects. So I want an object string. So I just this object. I have to provide a function that will manipulate that object. So I'm going to call that function transform. I have to call it filter. Now I can put this here to pipe my filter. Okay. So how do I create a string now? So I'm giving a function to this a transform function to this string. I have to do var filter equals function. What it's gonna get is an object from upstream that came from the Johnson string parse. I'm going to get an encoding, which I don't care for now, and I'm going to get the callback that I should use once I process my chunk of data. So I'm going to call this callback next. And so now what I can do is I'm not going to do anything to the object. I'm just going to let it through. I'm going to do this push object. So this will send the object I got downstream to the stream queue. And since I'm done with it, I'm going to call the next callback. So that means I finished. So actually, I still have the problem that I'm pushing an object to the next stream, which is the buffer stream. So what I have to do here is Johnson string define the object. Now my object is back to the string. So if I run this, I should still get exactly the same thing. I'm not very excited. Oops. Maybe.
In your PDF, you've only got callbacks to the next.
I changed the name here. The same. This push is right.
Line.
Well, I guess I have a stupid check or something.
Yeah, the code system arrived. You see the slides as a backtick instead of the single code.
Where?
So that's just yeah. Another request is the way to claim this function. If I do just function, I also remove the mark. It should not be. If you do function filter, that should work.
Yeah, so I was passing the variable here, so I set up a function.
Right.
So it doesn't work. So I need to pass a function here that gets called. So now I could do something useful with this. So I could do, let's say, object. Do something like this. Resolve this. I can do resolve or each. Get one result. And I'm going to split my results. Because I'm calling a function is to do this here, so I need to require this to give the context. Otherwise, I'll leave the context. And I have to solve here. So this should give me. Good. So now I'm getting, for each object that was in the array, that's what's getting piped out of this pipeline. So I could do something like if result study name match, let's say 1000 genomes project. Then only then push. So I get 2 results. So this is how I implemented the filter script. So you get access to this code, so you can try to do the same thing in the afternoon. I'll try to quickly move on. So, how much time do I have? Okay, so I'm just going to show quickly another thing. So one thing that might be a bit hard to understand with streams is how backpressure works. So our streams regulate the flow of data. So I have a code, example code for that. So I'm going to go quickly through it. So here in this code, what I'm doing is I'm installing a bunch of dependencies. I'm cleaning the terminal because I'm going to be doing some fancy stuff with the progress bar. I'm going to do 20 jobs. So I'm just going to create 20 empty objects and pipe them to the stream. And I'm going to have a counter that keeps track of progress as the data goes to my stream. And I'm going to have a fast stream and a slow stream. And this here is just the progress bars. All the progress bars will be rendered in the terminal using the module called multiplicator. So the relevant code for this is here. So I'm going to create a read stream using a module called streamify, and I'm going to give it an array. So an array of empty objects. That's going to be my read stream. And then I'm going to have 2 streams, the fast and the slow one. Then I'm going to apply the source to the fast to the slow. And because the slow isn't piping to anything, if I want to process the whole data, I have to call resume to make it read the whole data. Otherwise, it will stop as it stops fetching data from the upstream. And so the stream is going to be again a transform stream, and I'm passing an option. I'm passing this option called high watermark. So that's the size of my buffer. The default is 16, but I'm going to play with it so you can see what happens when I change it. And so then I have my transform function, object including next, just as I did in the previous example. Var self to keep track of the context. What this stream is going to do is going to execute command line tool, sleep in this case. So it's going to sleep for 1 second. It's the only thing it's going to do. And that tool gives me error, standard out, and standard error. But I don't care about it. I just implement the counter once I'm done waiting. And I push the empty object to the next stream so it can process it. And that's it. And the slow stream does exactly the same thing, but it sleeps 2 seconds instead of 1 second. So now I'm going to show what happens. So you'll have access to all this code, so you can play with it later. So if I do Node backpressure, what you see on the top is the fast stream processing jobs. So it's processing 1 job per second, and the slow stream 1 job per 2 seconds. So you see the slow stream is lagging behind, but that's fine because I have a buffer of 16, so the fast stream doesn't care. It's just moving forward. When I finish, the other one will still be processing stuff. Now, if I change the buffer size, let's say I put 4. 4 objects. If I run it again, so the fast stream starts going forward, but it will only be 4 objects in advance of the slow stream. So when it gets to a head, it will stop and wait for the slow stream to catch up, because it knows the buffer is only 4, so you cannot push any more data. So you can. That's how streams work, and that's why they're really cool to process the data. So if I change to 1, what will happen is this fast stream will go at the same speed as the slow one. So it does 1, and then the other does 1, and then it does 1, the other does 1. So even though the fast stream is fast, it only has to wait 1 second. It pauses to let the other one downstream finish what it's doing before pushing data. So you don't have access to this code. It's just sometimes a hard concept to grasp. I wanted to show this slide. I can now just quickly go through the concept of streams. I have some animations, maybe 2 minutes. So I wanted to show you how you code streams, because that's what we're going to do in the afternoon. But I know for some of you this might be a bit hard and a bit abstract, so I'm going to try to show how these concepts actually work. So streams, I implement a native construct in Node.js. They allow you to process data in chunks, so you can have readable streams, as you saw, which can be reading from a file, getting data from some service, or just the standard in of the tool. And then you pipe that with the pipe command to a transform stream, so a parser, a filter, or some crazy multi-thread analysis if you want to do. And then you can pipe to a write stream, which would be writing to a file or standard. Now, this is fine when your streams are fast and you don't have too much data. The data just flows in and there's no issue. Everything is fine, everything is perfect. Gets there, gets transformed. No issue here. But the problem is when you have a bottleneck. So one of the streams is slower. If you don't have too much data, that's fine. Even if the data takes a while to get processed, nothing will happen. But sometimes you cannot predict what would be, how small will be the stream or how slow will be the stream. And if you have a complex pipeline, you cannot predict where the bottleneck is happening. So the bottleneck got to be a network issue, network latency. So there's no way you can predict that. So your pipeline has to be robust to deal with that. So the first implementation of streams was what we call push streams. So they'll just push data downstream. So what would happen is if you have a big object, big chunk of data being processed downstream, when new data came in, it will get lost because it couldn't be piped to the downstream. So you end up, that's why you should avoid using stream, version 1 of streams, because it might lose data if you're not listening to that data. But now, the way streams are implemented, you can still use the push way, but the way you should do it is actually use the pull way. So downstream gives feedback upstream to pull more data. So what happens is new data comes in, and it's already full. When I do the this push data, it returns a false. So it tells automatically to the streams that you cannot push more data. So the content goes to a buffer. And if the buffer is full, it will just pause the upstream stream. Once I'm done with the data, so downstream I did this push data in the other two, and then to say I want more data, I use the callback. So in some of my demo I called next. You can call it whatever you want, but this function tells I want more data. I'm done with this. Give me more data. And you can only call the callback once, but you can call the push as many times as you want. So imagine you have a big chunk of data. And that's what I did in my example. I had this array, and I started pushing one object, each object of the array downstream. So you can break your chunk of data and do this push several times, as many times as you want. And once you're done processing that chunk, that's when you call the callback. And that's how streams can do things like backpressure. And so what you saw until now were read streams, transform streams, and write streams. But there's other kinds of streams. So duplex stream looks like a transform. You can write and you can read to it, but it's actually the combination of a writable and a readable stream. And they don't have to be anywhere related. They can be doing different things. You can imagine this as a server or a database. You can write data to it, you store it somewhere, and you can get data out of it. But these two streams, they're not connected. They just, the duplex is just an abstraction on top of that. And then another kind of streams you can have is the pass-through streams. So this is what you saw when I implemented the counters. So I had a stream that just counted what was coming in, and one didn't touch the data. So that's how you can implement metrics and see how much data is passing through. But the other useful thing that I use a lot pass-through streams for is that you can fork your streams. You have a stream that looks like a T, but it's actually not a fork. What's happening is you have your stream, your pass-through stream, that is just watching for the data coming in. And since it's watching it, you can then reuse that same stream somewhere else. So in my first talk, when I did the forks, I was piping the fork one to another stream. So that means then you can basically copy your data. So that might be useful if you're trying to do multi-thread, for example. Parallelization, you can do it that way by forking. If you're trying to query, now I have the same object duplicated, and I could do pass it to two different downstreams to do, for example, a different queries, like I was doing on SCBI, fetching all those data in different sources. And that's the basics of streams. So here I have a list of the benefits. We also sent you, last night, PDF with the details of streams. So as I mentioned, there's these 5 kinds of streams. And the way to implement it is usually use an abstraction, like for example, BCC. So I've been using, I showed you the transform streams, I use the module true to implement them. But module true is part of Mississippi. So you can actually just install in Mississippi and you have a bunch of tools. Or you can do it the hard way, and you just subclass the streams class from Node, and then you implement the appropriate methods. Be careful if you do that. You should actually install streams as a module too, because if you abstract the one that comes with Node, then you're locked in to that specific Node version. So if you don't want to be locked in, you just install the streams as a dependency, and then it's compatible with any Node version. So this, I think in the afternoon, this slide will be very useful because I have here how to create streams using Mississippi. If you go to the GitHub, you have code examples how to do this. And this is the easy way to do it, and that's what you guys should be doing in the afternoon. I put a bunch of other links. There's a bunch of, on the discussion, there's a bunch of useful stream modules you can use to do a bunch of cool stuff. These guys, Max, Matthias, and Substack, they contribute a lot of streams to Node.js. So if you follow them, you probably find a stream for whatever thing you're trying to do. And that's it. I hope that was all. Thank you. If you have any questions, please ask. Or come talk to me later.
So I'll keep you guys busy for another 10 minutes, not more, I promise. We have the lunch prepared, so 10 more minutes. We'll try to go quickly through what we want to do after we have lunch. And okay, cool. So don't panic if you didn't manage to follow up, like, or you fall out somewhere during the tutorial from Bruno. So we'll go back to all these concepts. We'll put links to all this documentation. I mean, most of you guys already know it. So you will, I mean, those things are more difficult. Some of them are more difficult to access. So hopefully by the end of the day, we'll talk more about them. So you'll grasp that. So what we want to do today, so where we want to focus from now on is basically three things. So in BioNode, we've already developed a module which is called BioNode NCBI. So we want a focus. We want a group of people to work on BioNode NCBI, and I'll explain what some of those things will be. So basically, we want to test the package. We want to kind of see how add some databases to it. We want to see how robust is it, and so on and so forth. We want to prototype new BioNode modules for new data sources, and I'll quickly tell you about some data sources. And we want to think about architecture challenges. So if you're excited about streams, and if you have some ideas on, I don't know, how we can use this type of streams, or how we can organize the data, work on the data flow, we want a group of people to work on that as well. So what data sources we want to work with today? So basically, we prepare a set of data sources. Those are not exclusive, but so I mean, if you have a data repository, a data source, they say like, I'm trying to get data from that for like more than 1 year. I want some programmatic API for that. Let's work on that. Cool, we'll do that. But we have, if there are no so desires, we have already put down a couple of them. So basically, we have the expression atlas, European variation atlas, the gene expression on the bus, database genotype, sequence archive. So basically, you will see here the repository name. So if you go to the BioNode hack organization, you'll find those repositories. Those are part of the BioNode. So NCBI, BioNode NCBI. And you can see that in BioNode NCBI, so this is kind of a meta repository. So there are a couple of databases. So you will get some information about them. So you have multiple organisms there. The DNAdigest is on the human, who is hosting it, the data type. So Charlotte showed you like all that plethora of different data types and access type. And we went to these repositories. We looked around and we found that some of them have, for example, APIs. So like REST APIs or APIs, or some way to extract the data. Some of them is basically HTML table in which says like gives you the link to files and so on. So when we see their table, it's expect we kind of see that table. So basically, what one will need to do is to scrape that file and that page and extract that information from that table. However, it might be that for some of these repositories where we say it has only API, might be that there is a table, or if we say the access type is table, might be that there is an API that we didn't find about it. So feel free to do that. So we have all those data sources. So another one is encode, cancer, so this is methylation data, mouse tumor biology from Jackson Lab, expression atlas, personal genome project, so this is one volunteer that are putting the genome cover. It's kind of interesting. Database the parent one, most of them. So you will find all these repositories in the organization. So you see this, but just want to make sure we really on the same page. So if you go to BioNode hack and you'll see the repository, and you go for each repository, you'll see, for example, BioNode DNAdigest, you have a README file which will give you as a lot of details about this, how to get the data, or to get the data, methods, various methods that they offer, the structure of the data. Some of them is more complete, for some of them it's less. You'll also find some examples of the response where there is an API. So what you will find in that description. So basically, in the README file. So there is a brief description and main URL of the data store, the data source. And then we try to answer a couple of questions. So, 3 questions. So you will find how to find the set of data sets that are available for that particular data source. And in some cases, you know, it's human, it's not relevant. Then how to request the information. So, you know, like some descriptions about the APIs. So that will be like 1, 2, 3. So how to get either one data set, or if it's a table, just grabbing all of them, or if it's a entity, some information related to that. Where to find the data sets, attributes, and how to get them. So, you know, like what each data set entity, what the object response that you get, how is that characterized, and then some information like accession number, URL, title, description, technology, and save that. So you'll find all this information for the repository, for all these repositories listed here. Now, if you have a repository, you say like, I want to work on that repository. My department has that repository, and I want to do that. We would love to start characterizing that repository following those steps. So, what do we want to achieve, or kind of some milestones during the day? So, the first goal, basically, the binary module. So, let's say somebody say, I want to work on the EVA. So, the first goal should be like a module that provides functions to get all the metadata, so the description of the data sets from their respective source. So, for example, I think this is EVA. So you will get an object like this, right? And then the repository contains a lot of data sets, so all of these. So basically, first goal, get those objects, right? Second goal,